I'm Ms. Newton, and I'm going to be talking today about the scientific method and how to properly fill out a lab report. The scientific method starts with questioning your surroundings, meaning if you're a scientist and you see something going on that you're curious about and you want to know more about it, you could do the scientific method. And you may find, after we talk about the scientific method, that you're already doing it frequently in your everyday life. So, examples of questions you may have as you're just going through your day. You might see an animal eating something or you might be curious about how something consumes food or whatever. And you may say, how does this organism eat? For example, my bearded dragon can drink water through its skin. So you might want to know more about that. Why does it live here? In other words, you see that animal, why does it live here? Is there a specific food it needs here? Is there a specific predator it's trying to stay away from? Why does it sleep during the day or night? And how many offspring does it give birth to at one time? So these are things that you might be curious about and you could further investigate. And there could also be questions just in your everyday life, like, why do I feel tired today? And then you can go through the day before and try to figure out, is there something I ate before bed? Did I drink caffeine too late in the day yesterday? Um, did I have to get up to use the restroom too many times? So you might try to figure out what was different about yesterday and how can I eliminate that tonight? And by eliminating something that might not allow you to sleep well, you're actually doing the scientific method. We'll talk more about that. So you're making observations right now. You may wonder about me, about if I'm nice, if this lecture is going to be interesting, and then you're going to find out by continuing to watch the lecture. Okay, after you ask your question, you can observe or perform an experiment. So in other words, it'll be time, well, you'd write your hypothesis, and then you would do an experiment. But an experiment doesn't always involve having two things and testing them. Sometimes it's just observing your environment and recording your data based on your observations. So for example, if you want to know how many birds visit your house each day, you could count for a period of time and then extrapolate that over a 24-hour period. So you could count for an hour, record how many birds come to your yard at that time, and then maybe compare it to a different day when it's cloudy. Maybe it was sunny the first day, it's cloudy another day, and see if there's a difference in the amount of birds that come. Now, by doing that, you're doing the scientific method by recording that data and observing and collecting data, but you're not doing an experiment. So you're not changing anything about your yard. We'll come to that. You're not, I don't know, blowing a whistle to attract them or scare them away. You're not doing anything different. You're just observing. So yes, we can do the scientific method by just observing. So then you could turn that into an experiment. If you want to know what kind of bird seed the birds at your house like, you could develop an experiment with 2 or more types of bird seed. So let's say you go to the store and you pick out 2 types of bird seed. You put both of them out in the same type of container, maybe in sort of the same location. And then you record how many birds come to each bowl. Or another way to record what the birds like best is by weighing the bird seed at the end of the day. If they ate more of this one, it's going to weigh less. If they ate less of this one, it's going to weigh more. So there's different ways to record data, but that is an experiment because you're comparing 2 things. So you can either do observational research or experimental research with the scientific method. So what is the definition of the scientific method? It's a process used to investigate the unknown. Unknown sounds really big and fancy, but it really just means, like in the case of your birds, you don't know what kind of birdseed they prefer, so you're going to find out. A process used to investigate the unknown. So there are generally 6 steps to the scientific method. Some teachers will break this step up into 2, and then there would be 7. But for the purposes of this class, we're going to just talk about 6 steps. And we've already talked about 1. Actually, we've talked about 2. The first step is observation. Now, sometimes that's not even something you try to do. You just happen to notice something, and something caught your attention. You observed something. The next step is to ask a question and maybe do some further research. You could go to Google, you could talk to people. Sometimes it's just posting something on social media and saying, hey, what brand of this food do you like? That's doing research. And then maybe later you'll test it by comparing it to different brands and tasting it yourself. So asking a question and doing research. The hypothesis is your prediction. How do you think this experiment is going to come out? Or how do you think your observation is going to go? The next step is to do an experiment, or like I said, record data while observing something. The next step is to record your data. And then the last step is to, well, I don't know if we would call this a step, but it's the last part of the lab report, is to write your conclusion or draw your conclusion, meaning, okay, what did I learn from this? And part of that conclusion is to write in your lab report, do the results support the hypothesis? In other words, I made this prediction. Did that prediction come through in the results of my experiment? And we'll talk about each of these in depth. Okay, here's another example of, I guess we would call this observational research, okay? And this is a real life example. There were some scientists that noticed that some frogs in a pond have extra limbs, meaning extra arms, extra legs. And they asked a question: why do so many frogs have a mutation or an extra limb? Is there something in their environment causing these defects, like a toxin? You may first research to see if the extra limbs in frogs is normal or indicates a mutation. Okay, so that is the observation, is that you see these frogs with extra limbs. The question is, why do they have extra limbs? Could it be this? Could it be that? What's going on? And you may research, you may look into it, see if this is common in that area or something. Next, you would develop a hypothesis based on, let's say, this frog experiment. So you're basically proposing an explanation as to why this is happening. So a hypothesis is a proposed explanation that tries to explain an observation. A hypothesis should be written so that it can, I'm just going to start with number 4, so that it can provide a yes or no answer. And we'll talk about that in a minute. The hypothesis should be able to be tested using an experiment. It should be able to be proven wrong. It should provide measurable results, like specific numbers, and provide a yes or no answer. So I'm going to give I do have an example. Here's an example of a hypothesis for these extra limbs on the frogs. The number of frogs with extra limbs in 5 ponds that are polluted with chemical X is higher than the number of frogs with extra limbs in 5 ponds without chemical X. So this hypothesis is guessing that there's a chemical causing more frogs to have extra limbs. So they're saying there's going to be, when we do this observational research, we are going to see more frogs with extra limbs in this pond that has more chemicals. And we're going to see less in this pond. So can that be answered with a yes or no answer? Yeah, because they're going to count the number of frogs. There's going to be definite numbers, and they can say, yes, there are more frogs with extra limbs in the pond with a chemical, or no, there are not. Is it measurable results? Yes, because you can count. So measuring means, counting, anything where you're recording data with numbers. Can it be proven wrong? Yes, it can be proven wrong that, that you're thinking that it's the chemicals. And can it be tested using an experiment? Now, in this situation, it's not an experiment where they're doing anything to these frogs, but they are going to be checking the chemical levels in the ponds. And for the most part, we're going to consider this an observational research or an observational experiment. So that's the hypothesis. Now, when this is not observational and you're actually creating an experiment, you can use an if-then statement. Okay, here's an example. If I give this dog food A and food B, then the dog will prefer food A. That's my hypothesis. So it's an if-then statement. If I give this dog food A and food B, then the dog will prefer food A. Now, when you're actually writing out a hypothesis, you're gonna say what the food is. You could say chicken and beef. You could say wet food and dry food. I was giving an example and I couldn't think of foods right then, but that would be another example. So if I do this, then this will happen, okay? That's another example of a hypothesis. All right, so then we have the experiment section. You record the data collected from all experimental groups. Report the data in a chart. Oops, wait a minute. Let's go back. The experiment, you would measure the amount of chemical X in all of the ponds, count the healthy frogs and frogs with extra limbs, and compare the ponds. Your experiment consists mainly of observation and collecting data. So that's the experiment. Now let's go back to my dog food experiment. Well, in my lab report, I would write down the supplies I'm using. So bowls, I would say 2 bowls, food A, food B. Maybe I'm going to weigh the food afterwards to see how much of the food the dog ate. So maybe I'm going to include a scale on that list. So I'm going to write down the materials used. Then I'm going to write down the procedures. So in that example, the procedure would be, I scooped, or not I would say scoop 3 grams of food A into bowl A. Scoop 3 grams of food B into bowl B. Place in front of dog. Now at this point, you could say something like, give the dog 5 minutes to finish eating, then take the food and weigh both of the foods to see how much of each the dog ate, okay? Or you could record which food the dog goes to first, but I think weighing the food is probably going to be the best way to do it. Now, sometimes people will do their experiment 3 times because maybe that day that you did the experiment, the dog was like tired and didn't feel like trying both foods. It just ate the first food it came to, or, there could be something going on. So if you try that experiment on 3 different days, you could kind of combine your data and come up with a better conclusion or better results. Okay, so the next step is to record your data. Record the data collected from all experimental groups. Record the data in a chart or graph if possible. So in the situation with the frogs, they counted how many frogs in each pond had the extra limbs. They put this in a chart. So pond A, they had the number of extra limbs. Pond B, they had the number of extra limbs. In the situation of the dog food experiment, you could do, trial 1, trial 2, trial 3 in a chart and put the measurements of food A and B for each trial, each time you did the experiment. And when it says collected from all experimental groups, with the dog food, that means the experiment is comparing 2 different type of foods. So you're going to record the data from food A and food B, how much each one weighs afterwards. So those are the experimental groups. Next, you're going to analyze your data and draw your conclusion. So we've already gotten the results. We've written them down. Maybe we've put them in a graph. Now we're going to sit and look at it. We're going to think about what the trend is, what we saw. And we're going to draw our conclusion. So your results show that the chemical levels in the 2 sets of ponds are different, but the number of frogs with extra limbs is about the same. Analyze the data to see if it supports or disproves your hypothesis. Your results with the frogs show that your hypothesis is false. What do you do next? Okay, so actually in this actual study, they did find out that there were not more extra limbs in the pond with the chemical. So the hypothesis was wrong because they predicted that there would be more frogs with extra limbs in the pond with a chemical, but it wasn't that way. They were all the same, both ponds. Same number of frogs with limbs, or very, very close to the same number. So what do you do next? Do you just say, it's bad data. I'm going to throw this whole thing in the trash can and I failed. I failed my experiment. No, you do not do that. So what do you do if your hypothesis is proven wrong? You can publish your research if you're a scientist and you're actually going to write this into a big, professional lab report. You're going to publish your research and that's it. You're done. And then and we'll come back to that. The other thing you can do is develop another hypothesis, something completely different, and perform another experiment. Okay, so in the situation with the frogs, let's say you did not prove anything. So it's not the chemicals, it's something else. You could publish your research because that helps future scientists who want to study this to see what scientific experiments have already been done. So then they can go and do something else. So they might say, oh, look, we found this research article. It's not the chemicals. So we're not going to study the chemical aspect again. We're going to study something else. So that really does help scientists to build on that research later. And then, you can develop a hypothesis. So in this situation, a new hypothesis. In this situation, you might say, okay, it's not chemicals. Let's see, what else could it be? Okay, maybe there's a certain gene pool going on here, like certain genes that got into this frog population. And we're now going to study the DNA or something. Okay, so then you're going a whole different angle. Now we're going to set up our experiment this way. And you could do a whole new experiment or observation or something. So even when scientific experiments turn up to be not what you expected, they're still valuable. Okay, so we're going to talk more about experimental design, setting up two parts of an experiment. Okay, we talked about the dog food with the two foods. We talked about the bird seeds with the two types of seeds. We can also, I think for this example, I'll use pharmaceutical medications. When a pharmaceutical company is studying a new medication, they're going to have a large group of people for, let's just say, I'm just going to pick some numbers. Let's just say 100 in one group, 100 people in another. They're going to give 100 people this new medication that they're studying, and they're going to give 100 people a placebo pill, which means it's a pill. They don't know whether or not there's medicine in it, but there's not. So the placebo pill is like a sugar pill. There's no medicine in it. It looks like medicine. They made it look like medicine, but there isn't. So these two groups of people, if it's called a double-blind study, these two groups of people don't know if they're taking the medicine or the placebo. Nobody tells them. So they're blind. Okay, that's where the blind word comes in. Double-blind means the participants don't know who's on the medicine, and the doctors recording the data don't know who's on the medicine. It will be later revealed. So that's called double-blind research, and that's considered very good scientific experimental, the way it's set up. It's good design because it helps for the patients to not say, oh, I'm taking this pill and I'm feeling this way, so it must be the medicine. They're not going to be as likely to do that, and the doctors as well, they won't be as biased. Okay, so the control group, that's the part of the experiment that does not change. And ignore this right now, we're going to come back to that. The experiment that does not change, or part of the experiment that does not change with the medicine, is the placebo. Nothing changed in these people's lives, except that they're taking a sugar pill and it's not going to affect them. That's the control group of people. The placebo group is the control group. The experiment group is the part of the experiment that does change, the group of people that does take a new medicine. Okay, so that's the part of the experiment that does change. Now, there is an experiment, another example is an experiment that the Mythbusters did, where they had a car, a regular car, and then they had a car with dimples poked out, not poked out, or dimples like think of a golf ball that have these little like dimples in them all over the golf ball. They did that to the car. So they had a regular car and a dimpled car like a golf ball. And they tested the gas mileage. So they put gas in the car, they had it go a certain period of time or a certain distance, and then they saw how much gas was left. So they wanted to see if the dimpled car got better gas mileage or the regular car. And I actually don't know the answer to that. We don't need to know the answer to understand what I'm about to tell you. If you want, you can go to Mythbusters and type in, or go to YouTube, type in Mythbusters, dimpled car versus regular car, or something like that, and you'll probably see it. But the regular car was the control part of the experiment, the car that does not have dimples. So they're comparing, they're using this as a regular car. The other one, the experimental car, has dimples because they wanted to see if they could get better gas mileage. So they're using the control to compare to a regular car. They're using the dimpled car as the experiment. And that way they have something to compare it to. And they were identical cars, except for the dimples on the one car. So control, experiment. Okay, so now we're going to go even further and talk about the independent variable and the dependent variable. And I'm going to talk about a few of the Mythbusters experiments. They always do big, giant experiments. They don't just stand at a desk and do stuff. They go all out, okay? I'll talk about 1, 2, and 4 because I know the most about those. All right, so independent variable, dependent variable. So there's two groups. The independent variable is the difference between the control and the experiment groups. The variable, this is the variable changed by the scientists. So what did they change? They changed the dimples. So the independent variable is whether or not the car had dimples or not. Okay. The dependent variable is what is measured. What are they writing down? What are they recording during the experiment? This is the data collected from the experiment. In the dimpled versus the non-dimpled car, they were recording the gas mileage. So the dependent variable is the gas mileage. Okay, let's look. Actually, I don't know how to explain this one either, but you could look this up. They had elephants and they had mice. And they put the mice somehow inside a cup that was also stuck inside the elephant dung. I don't know. That one's not that easy for me to explain, but the elephant's reaction to the mouse was what they were recording. Did this elephant react when the mouse was there? Did it react more when the mouse was there versus no mouse? Okay. They also, and I really have always wondered about this. They also experimented with people walking or running through the rain. So like when I go, park at a store and it starts pouring and I don't have an umbrella and I really need to go into the store, I may ask myself, should I just walk into the store or should I run? Which one will result in less water on my clothes and me being less miserable in this air-conditioned store, all wet? I don't know the answer to that. And again, I don't know the result of that experiment, okay? I only know how they set it up because all I have to teach you is the variables. You can go look that up. So you would go to YouTube and type in Mythbusters, people moving in the rain. Maybe that'll help. Or type in Mythbusters, independent and dependent variable. That's probably going to help you find the video. Okay, so in this example, they had a group of people who walked through the rain, and they had a group of people who ran through the rain, and they did it at the same time so that it was the same amount of rain coming down. But so walking versus running. So the independent variable, the variable changed by the scientist was the speed that they went. One group walked, one group ran. So the speed, that's the difference between the two groups. The dependent variable was the mass or the weight of the rain on their clothes. That's what they were measuring. They literally measured the weight of the rain on their clothes when they were done. Now, I don't know if they had them change their clothes and they measured the actual clothes they were wearing in both groups, or if they just measured the people before and after, and assuming they weigh more when their clothes are more wet. I'm not sure how they did that. The problem I see with that is, well, maybe you were sweating when you were running, so you have even more sweat weighing you down. But then again, no, that wouldn't affect it. So I don't know exactly how they pulled that off, but they weighed the clothes to figure out the exact mass of rain on their clothes. So in that case, it's what they measured during the experiment. They put an actual amount, this group, their clothes weighed, I don't know, 2.5 grams. This group, their clothes weighed 2.1 grams, so they had an actual number. And they were able to see how running or walking affected how much rain got on your clothes. Okay, so that's the independent variable, the variable changed by the scientist. The dependent variable, the data collected by the scientists. What are they measuring? And that is it. I hope you enjoyed that, and I will talk to you soon.